Hello class, a very warm good afternoon to you all. Myself Vivek Prakash Yadav and I am here to take your biology class, the class 11th. And in our continuous series of class, we are talking about photosynthesis, right? And in today's class, we are going to talk about chemiosmotic hypothesis. Okay, we are going to talk about chemiosmotic hypothesis. So what is our topic today is chapter is photosynthesis. Now in this chapter of photosynthesis, what we are going to discuss is chemiosmotic hypothesis. Okay, so today's topic is chemiosmotic hypothesis. So we need to know actually what is this. See, as we have studied already that in photosynthesis we have discussed about cyclic and non-cyclic photophosphorylation. So what is photophosphorylation? I told you in the last class that is photophosphorylation is the use of light energy for the production of energy. Okay, use of light energy for the production of energy. Now in chemiosmotic hypothesis actually we are going to study the mechanism. Okay, we are going to study the mechanism how ATP or we can say NADP plus, okay, means energy molecule. Okay, mechanism how ATP and NADP plus are produced. Okay, it is very much similar. Okay, it is very much similar to the process of respiration. Okay, it is very much similar to the process of respiration. Where does it take place? In mitochondria. Okay, in mitochondria. But what is the difference? What is the difference in chemiosmotic and the respiration? Actually, what happens when we talk about mitochondria? In the case of mitochondria, the proton gradient, okay, the proton gradient, that is hydrogen ion formation, is formed outside the lumen. Is in the outside the lumen, and the entry is inside. But when we are going to talk about this, that is chemiosmotic hypothesis, in the case of plant, it takes place in chloroplast. Yes, it takes place in chloroplast. Okay, and we all know that chloroplast is a double membrane bound structure. Here what happened? The proton gradient is actually inside the lumen. The proton gradient is actually inside the lumen is higher. Okay, inside the lumen is higher. So the breaking down of the proton gradient, okay, the breaking down of the proton gradient would be from inside to outside, from the inside to the outside. So here we have to discuss that what are those enzymes which are present in the membrane, okay, for the production of NADP and also for the ATP. Okay, so we will discuss in detail about this, but just I want to inform you about the introduction thing. Now first of all we are going to draw a diagram. Okay, first of all we are going to draw the diagram, and then we will discuss. See, proton gradient is also an electron. The continuous supply of electron is also necessary, and we all know how it which provide continuous supply of electrons. In the non-cyclic photophosphorylation or in the jet scheme, I told you that in photosystem 2, which is actually in the membrane of stroma lamella, yes, stroma lamella, the photosystem 2 is there, and photosystem 2 actively participate in the splitting of water, okay, splitting of water, and when water molecules are going to be split, there is release of electron, and that system provide continuous supply of electron, okay. With that, we will also discuss the other thing. Now we are going to draw a diagram. See, when you are going to draw the structure of the chloroplast in the of the grain, chloroplast, you see this is the okay. It is a double membrane structure, we all know. Okay, we are going to draw a diagram for the chemiosmotic, how ATP is going to be synthesized. Now, I told you that there is presence of photosystem 2 and photosystem 1, both are going to be present in stroma lamella in the membrane. Okay, it is going to be present in membrane. So, here you can see this is photosystem 2, and there are light acceptor or electron acceptors, that is known as light of E680 nanometer. Yes, that light of E680 nanometer going to be absorbed in photosystem 2, and in photosystem 1, we can say photosystem 1, there is light of E700 nanometer, means red light of 700 nanometer is going to be absorbed. Now this inner structure, okay, this inner structure is your lumen side, you can say lumen, and external one is your stroma, okay, stroma. Now in photosystem 2, as I told you, what happened, the splitting of water molecules takes place. Yes, when light energy getting trapped, they are causing the splitting of water molecule, okay, plus free oxygen. Okay, this is going to be released. Okay, half O2 plus oxygen, hydrogen, and your 2 electrons going to be released here. Okay, now what happening, that the hydrogen which is produced here, okay, which is going to act as a continuous supply of proton, it is going to be developed proton gradient. Okay, proton gradient hydrogen. So this is the reason why the proton gradient is excess or inside the lumen, but in the case of respiration actually it is outside, not inside the lumen. Okay, now there are proton 1, I told you that photosystem 2 and photosystem 1 when together they participate, they form jet stream. In between, what happens when light energy fall, okay, when light energy fall, what happens, the electron get excited, okay, and there are electron acceptors, which are present, that is known as cytochromes. Okay, cytochrome B6, cytochrome, I told you there are electron carrier or acceptor which are known as cytochrome. Okay, and from they are going to be carrying the acceptors, then cytochrome is going to carry them to the photosystem 1. Now there what happened, I told you that when electron get excited, Okay, when the electron gets excited, they move upward movement, and due to redox reaction, downward process takes place, and there is release of or the formation of ATP takes place. So in between, the one which is going to be accept the electron is plastoquinone. Plastoquinone is an electron acceptor, okay, that is going to accept, and cytochrome, which is electron carrier, they are going to carry those electrons to the photosystem 1. There again light fall and get excited, again down the process takes place during jet stream, okay. And what happened in the membrane? There is NADP reductase, okay. Here there is an enzyme that is called NADP, okay, NADP reductase. NADP reductase enzyme is present. Okay, these enzymes cause conversion of NADP plus to NADPH plus hydrogen ion. So in this way, NADP is going to be means in case of continuous jet stream. Okay, what happened due to the enzyme NADP reductase which is present in the stroma, that is in the membrane facing. See that one NADPH1 is facing towards the stroma side, okay, and which is used, the NADP which is used, that is not going to be stored, that is actually immediately get consumed for the conversion or for the reduction of carbohydrate to the glucose, carbon dioxide. I mean to say the carbon dioxide reduction during photosynthesis, the reduction of carbon dioxide to the glucose takes place. So during that time, during that conversion, the energy required is actually obtained from that NADP. So NADP is going to form NADP. It is ultimately or immediately get consumed. Okay. Now what happens? As we can see, due to photophosphoryl, due to splitting of water molecules, okay, due to splitting of water molecules and due to excitation of the electron, what happened? This is the plastoquinone, which act as electron acceptors, okay, excited electron acceptor. Due to this also, hydrogen ion entry inside the lumen takes place. Okay, one source is through the splitting of water molecules, and another source is plastoquinone, which is causing the entry of proton, that is hydrogen ion. As a result, what happens inside the lumen, the proton gradient is getting increased. Now there is one more enzyme, ATP reductase. Okay, there is one more enzyme that is known as ATP reductase. Okay, now they have ATP reductase. This enzyme, just a minute. This enzyme is divided into two parts, that is chloroplast F0 and chloroplast, you can say, F1. Okay, these two parts they have classified, they are also embedded in the membrane of the granum. Okay, now what happened? See, we are talking about proton gradient because actually it is the breaking of proton gradient. Okay, it is the breaking of proton gradient which is going to produce or which is going to form the ATP. So what happened? Inside the lumen, the concentration, the gradient of proton is getting increased. Now what will be happen? This enzyme ATP synthase, okay, ATP synthase, the CF0 region. This region is actually responsible for the entry of this proton gradient or hydrogen ion, and when they come to the CF1 region, okay, CF1 region, that hydrogen ion or the proton, which is proton gradient, convert in the form of ADP, okay, convert in the form of, when they are getting inside, it is actually the release of ADP. This hydrogen ion cause the addition of what? ADP plus inorganic phosphate, okay, ADP and inorganic phosphate. So what happened when this proton is getting inside through this channel, that is F0 and comes out through the CF1 particles, okay, F1 particles. So what happened? this hydrogen ion or proton gradient help the conversion of ADP, that is adenosine diphosphate. Okay, conversion of ADP, that is adenosine diphosphate, and help with the addition of inorganic phosphate. So what happens actually when this ADP, adenosine diphosphate plus inorganic phosphate combine? So what will be happen? The bond energy start forming. When they are going, they will form ATP, that is adenosine triphosphate, and initially it was diphosphate. So the phosphate molecules when get joined with diphosphate bond, so this bond means the energy that we are talking, the formation or synthesis of energy in the chloroplast that we are talking, actually that is getting accumulated in the form of bond energy. Okay, that is getting accumulated in the form of bond energy. That energy when break down, it release energy to perform the different kinds of activity that need to be participated, that going to be occur during the process of photosynthesis. So this is what actually chemiosmotic hypothesis. I mean to say, when the photosynthesis takes place, the electron transfer, the cyclic, non-cyclic photophosphorylation that we used to say for the production of ATP and NADP as energy molecules. So what is the mechanism actually working behind that? How the membrane, how the lumen participate in the formation of that energy. So I told you, one is, see, in the case of cyclic there is formation of only ATP, okay, ATP, no NADP, yes, because after excited, when in the cyclic photophosphorylation, I told you in the last class that when electron get excited, okay, after harvesting the light, okay, trapping the light, that does not transfer to the energy, but again by the electron acceptor, they again retransfer to the light harvesting complex. So during that process, when the excited electrons are accepted by electron acceptors carrying back to the light harvesting complex, only ATP form. But in the case of non-cyclic photophosphorylation, first time at the photosystem 2 electron excited, they get carried, okay, again they with the help of electron carrier, that is cytochrome, they carry to the light harvesting complex 1, or we can say photosystem 1. They are again light of 700 nanometer strike, make them excited, again they excited, and then down redox reaction takes place, and downhill process bring them to the NADP molecules, energy molecules. So this is the sequence of the process of formation, and this NADP, which is form energy molecules, it gets immediately used by the process or during the photosynthesis for the conversion or for the reduction of carbon dioxide to the carbohydrate or glucose, which is stored in the form of starch. And what about ATP that is produced from the chloroplast, okay, during photosynthesis, is mainly because of this proton gradient. And what is the source of this proton? First, the photosystem 2, where the splitting of water molecules takes place, as a result the hydrogen ion getting produced. Second thing, in the photosystem 2, when light strike and the electron get excited, so the electron are get accepted by the electron acceptors like plastoquinone. They also help the release or transfer of the hydrogen ion or proton inside. It means proton pump, we can say they also act as a proton pump to make the entry of the proton inside the lumen. As a result, what happening? The proton getting increased, and the ATP synthase enzyme is there which acts as a channel to break down of the proton gradient, and from when the proton gradient is coming out from that, what will be happen? The adenosine diphosphate and inorganic phosphate getting attached, means the energy, proton energy is getting consumed in the form of bond energy, and ATP is formed. So hope you people have liked the class, and with that we are going to stop today, and yes, in the next class again we will continue with this and other topics. Okay, so with this, thank you and have a nice day.